Directions for questions 5 to 8. The passage talks about the relationship between fever and bacterial infection. Now, initially it says that it has long been known that serum iron is essential for bacterium to multiply. Serum iron means iron that is there in the blood. And a particular scientist named Garibaldi first suggested a relationship between fever and iron. What he found out is that microbial synthesis of siderophores. Now, siderophores are substances that bind iron. That is, bacteria use siderophores to bind iron or acquire iron from the blood. And microbial synthesis of siderophores in bacteria of the genus means type, of the type Salmonella declined at environmental temperatures above 37 degrees Celsius and stopped at 40.3 degrees Celsius. So at high temperatures, bacteria are not able to produce siderophores, and it is with the help of these siderophores that they are able to bind iron or acquire the iron. Thus, fever would make it more difficult for an infecting bacterium to acquire iron and thus multiply. It becomes difficult to multiply because they are not able to acquire iron. Cold-blooded animals were used to test this hypothesis because their body temperatures can be controlled in the laboratory. That is, we can easily control the temperature of cold-blooded animals. If you increase the environmental temperature, then their body temperature will also increase. But for warm-blooded animals, they have a constant temperature. For us human beings, the temperature is 37 degrees Celsius. We cannot change that temperature easily. That is for warm-blooded animals. For cold-blooded animals, we can easily control their body temperature. And a particular scientist named Kluger, he made a study on animals called iguanas. They were infected with potentially lethal bacterium A. hydrophilia. And what was found out that at a higher temperature of 42 degrees Celsius, they had a higher survivability rate. But at the higher temperature of 42 degrees Celsius, when the animals were injected with an iron solution, then the mortality rates increased drastically. So this was what was found in cold-blooded animals. Now, research to determine whether similar phenomena occur in warm-blooded animals is sorely needed. That is, further study has to be done on warm-blooded animals also. So let us start with the questions now. The passage is primarily concerned with attempts to determine what? The role of siderophores in the synthesis of serum iron. Siderophores are used to acquire iron or bind iron. It is not used in the synthesis of iron. So A is wrong. Let us look at option B. New treatments for infections that are caused by hydrophilia. No, that is wrong. The function of fever in animals. That is the right answer. That is, general study is how fever affects bacterial infection. So option C is the right answer. According to the passage, Garibaldi determined which of the following. Now, what Garibaldi determined is that the bacteria need siderophores to acquire or bind iron. And at higher temperatures, bacteria are not able to produce siderophores. That was what was determined by Garibaldi. Now look at option D. There is a relationship between synthesis of siderophores in bacteria of the type Salmonella and environmental temperature. That is what he found. That is, at higher temperatures, the bacteria of genus Salmonella, they were not able to produce siderophores. So there's a relationship between synthesis of siderophores and environmental temperature. Hence D is the right answer. Which of the following can be inferred about warm-blooded animals solely on the basis of information in the passage? In warm-blooded animals, we have a constant temperature. That is, in humans, it is 37 degrees Celsius. So they have a constant temperature and we cannot control that temperature that easily. But in cold-blooded animals, we can easily control their body temperature. So look at option A. The body temperatures of warm-blooded animals cannot be easily controlled in the laboratory. That is the right answer. So A is the right answer. If it were determined that similar phenomenon occur in warm-blooded animals, which of the following, assuming each is possible, is likely to be the most effective treatment for warm-blooded animals? Administering a medication that lowers the animal's body temperature. That is wrong. Actually, we would prefer to increase the body's temperature. So A is wrong. Injecting the animals with an iron solution. That is wrong. That is, we must make the iron unavailable to the bacteria. So injecting the animals with iron solution will actually increase the mortality rate. So B is wrong. Administering a medication that makes serum iron unavailable to the bacteria. That is the right answer. Hence C is the right answer.